hemoglobin matrix. It transports vital substances between body cells and the internal environment. It maintains stability of interstitial fluids. Okay, what that means is it supports your homeostasis in your body. Because of blood, it keeps your body at 98.6, and that would be what we call homeostasis. It distributes heat. Blood varies with body size. It changes in fluid concentrations, changes in electrolyte concentrations, and the amount of adipose tissue. Blood is about 8% of your body's weight. The adult blood volume is about 4 to 5 liters in females and 5 to 6 liters in male. Make sure you know that bullet point, and make sure that you know that blood maintains stability and interstitial fluids to support homeostasis. Blood cells form mostly in your red bone marrow. So this would be red and your white blood cells. They are formed due to homeostasis, and when your body needs more blood cells produced, then your blood cells produced out of your red bone marrow, out of your bones. We'll talk more about that down the road. As you get older and that red bone marrow turns into yellow bone marrow, then your red blood cells do not produce as much and as often. That's part of the aging process, part of dying. So we have what we call red blood cells, RBCs, white blood cells, and platelets. Platelets are fragments. Blood cells break apart and they become fragments, okay? And so eventually, blood cells, especially red blood cells, after about 120 days, they're no longer in existence. So what happens to them? Well, the fragments are basically recycled inside of your liver. And so your liver disposes of red blood cell, and so eventually ends out your fecal matter in your urine. That's why urine has kind of a bloody color to the urine. It makes it that straw color. We call these cytoplasmic fragments or platelets. That's another name. So no cell fragments are also known as cytoplasmic fragments. This is interesting here. It shows you your blood composition. Notice that your blood breaks down into either plasma or what we call formed elements. Okay? Plasma is your nutrients. 55% of the volume of blood is plasma, okay? So your elements are less than your plasma. So you have more in plasma, and those are all your nutrients. Proteins, gases, water, electrolytes. But your elements would be either red blood cells, white blood cells, and platelets. They all three make up your formed elements. And then your white blood cells class break down into 5 different types. White blood cells' main job is for defense. Red blood cells' main job is to carry oxygen to your cells. Then your proteins inside of your plasma, albumins, globulins, and fibrinogen, all is part of helping clot when there are tears or breakages. So they even break it down by percentages. You will need to know those percentages, okay? Right now, I want you to know plasma is 55% of the composition and formed elements are 45%. And then know what plasma is made up of, and then know what formed elements are made up of. Okay, so the components of a blood sample. So if we took a blood sample and we come up with this composition, plasma would be the largest amount, right? At 55%, it's going to be straw-colored fluid matrix of blood. Your blood cells are going to be around 35 to 46% in females, 40 to 54% in males, okay? So then red blood cells are going to come up somewhere around 45% on average. Well, let me say that back. The formed elements are going to be 45%. That makes the 100%. But remember, out of that 45%, red blood cells are going to be 35 to 46 in females and 40 to 54 in males. They're round in the bottom of the tube, or they're found in the bottom of the tube. Red blood cells, of course, round. Percentage of red blood cells is called hematocrits or HCTs, and then your white blood cells really only make up about 1%. And you think, wow, all those different white blood cells and their job that they do only make 1% of the volume of blood. Yes, so formed elements, remember, the majority is red blood cells, and their main job is carrying oxygen. But white blood cells is only about 1%, and it's a buffy coat. And let's see if we can see that on the next. So plasma is 55%, red blood cells is somewhere, or is more of what we say 35 to 50%, comes in around 45 as average. And that buffy coat is about 1% of your white blood cells. And keep in mind, we have fragments as well, which don't really even register. Universal precautions, a set of specific safety measurements taken by healthcare workers to prevent the transmission of bloodborne infection pathogens. That's why when you go to have blood drawn, there are gloves, there's precautions taken. They seal off the tubes and dispose of the needles. It's estimated that 4 to 7% of new cases of infectious diseases are transmitted by unsafe injections. 4 to 7%. Is that going to be higher in third world countries? Sure. Is it going to be less in developed countries like the United States? Yes, it's because of it's the amount of the supplies and also just the safety protocol that's required. Personal protective equipment, gloves and masks. Use of fume hoods, sharps containers, safe workplace practices such as hand washing, all follow in this. Okay, so the origin of blood cells, what we call, so we're going to start with formed elements. And those formed elements, remember, break down into red and white blood cells. So hematopoiesis, I might be saying that right, I might not be saying that exactly right. What it means is blood cells originate in red marrow from hemocytoblast, which are types of stem cells. I want you to know that stem cells can divide, and so what we do with stem cells, and stem cells are specialized cells. And we think about stem cells being specialized where cancer patients who have blood cancer, they might give them blood infusions and try to get their stem cells to these specialized cells to divide on a cancer patient to get rid of the pathogen that's in the blood, whether it's cancer from leukemia or multiple, let's see, what is it called, multiple myeloma. These are kinds where then they'll do blood infusions and they'll try to get those stem cells dividing and eradicate the cancer. Sometimes it works, sometimes it don't. They call this lymphoid stem cells and myeloid stem cells. These new cells respond to growth factors, ultimately called hematopoietic growth factors. Lymphoid stem cells give rise to lymphocytes. So lymphocytes are white blood cells, right? So myeloid stem cells give rise to other types of formed elements, red blood cells. So I really want you to know that. The difference between the stem cells, one is for white blood cells, that's lymphocytes, and for red blood cells, that's myeloid. And that's important because if a person has a white blood cell count that's really high, chances are this person has a blood cancer, right? So in the medical field, people that have this in their family history, chronic leukemia can attack older people. That's why they call it chronic. Acute leukemia are these kids that have to fight it from day one. It's deadly. But the technology is so much better so remember, lymphoid stem cells are dealing with red blood cells. So what are you talking about? When someone gets a disease, let's say they get HIV, well, their white blood cell will drop below normal. It won't be high and elevated. It might drop below normal, which tells a medical profession that, hey, there's something going on here. The white blood cell count is not high, it's too low. And so know that. So here you can see blood cells where it talks about the stemloid cells and the breakdown from myeloid and lymphoid. Go through and know which are which. And remember, white blood are going to be under lymphoid and myeloid is going to be under platelets and erythrocytes, which erythrocytes are red blood cells. Just keep that in mind, class. So you just now learned a new word, erythrocytes is red blood cells. Always know that. They're biconcave, disc-shaped. They consist of 1/3 of the hemoglobin. Okay? So, you remember me telling you about some SACS questions? I want you to know that red blood cells are biconcave disc-shaped. That means they're like take a frisbee, flip it upside down where the rim of the frisbee is sticking up. That's a great example of biconcave. And that means it holds the oxygen inside of that concave disc shape. So know that for the SACS question. Also know that your red blood cells, drop one down, lack nuclei and mitochondria. Most people think all of our cells have a nucleus and a mitochondria. Well, they do. All of our trillions of cells except for our blood cells, red blood cells. So I want you to know that for SACS question. They're bright red because of the oxyhemoglobin. Oxyhemoglobin carries the red color of red blood cells. So when oxygen hits blood, it's oxyhemoglobin. When there is no oxygen, it's deoxyhemoglobin. Okay? And it gives you a bluish color inside of might be able to look through transparent through your skin and see a bluish tint. That is deoxyhemoglobin. blood cells cannot divide, class, only your stem cells. Blood cells cannot divide because they can't synthesize protein. Even though protein's in the blood cells, it can't manufacture protein. Therefore, it cannot divide. It can produce ATP through glycolysis. So your blood can get its energy without oxygen through glycolysis by help from the liver. Okay? We talked about the liver being an oxygen bank in bile 137. Remember that? Okay. Mature red blood cells produce ATP through glycolysis. They do not use oxygen because they lack a mitochondrion. Your spleen and your liver removes worn out blood cells. They recycle them. So your bloodiest organs is your liver and your spleen. Here's the structure, as you can see. They're about, from the top view, the width is 7.5 micrometers, and their depth is about 2.0 micrometers. Here is your red blood cell count that I'd like for you to know. For males, the typical range, this is in microliters, it's 4,700,000 to 6,100,000 microliters. In adult females, it's 4,200,000 microliters to 5.4 million. And in children, it's 4.5 million to 5.1 million microliters. Red blood cells count are useful in the diagnosis of diseases and elevation of the progress. The changes in red blood cell counts reflect changes in blood oxygen carrying capacity. Here's a new term, erythropoiesis. That means red blood cell formation. We know then that red blood cells come from bone marrow. So after birth, red blood cells are produced exclusively by tissue lining in the spaces in your bones. Certain bones produce more red blood cells than others, but there's a space or a lining in the bones that allows this process to take place. It occurs in your red bone marrow. Low blood oxygen causes the kidneys and liver to release a hormone known as EPO. This is erythropoietin, which stimulates red blood cell production. So if your red blood cells start to get low, EPO is produced, then red blood cells are being synthesized. So a negative feedback mechanism means that it continues, it's a good thing, right? Your body continues to produce red blood cells, okay? Remember I said about 120 days. So that's a good thing, that's why they call it a negative feedback. This goes on your entire lifetime. It just slows down as you get older in your senior years because your red bone marrow turns to basically yellow bone marrow, turns to fat. Your liver starts turning to fat. Your spleen is just about done, you know, if it's even a spleen left. So within a few days, many new red blood cells appear in your blood. So, test question here for you. Athletes abuse EPO because it increases the red blood cell supply which brings more oxygen to the muscles. You all heard me talk about Bruce Jenner. Bruce Jenner is one of the most disgraced United States athletes in the world. He's known because he cheated. For years, he doped up with EPO, and he would have a competitive edge, and he would just, these Tour de France's that he would do, he'd just wipe people out. For years he did this, in his prime. Everybody wanted to be with him, and then technology caught up with him. So why didn't technology catch up with him 10 years ago or 15 years ago when this was going on? Well, at that time, EPO does not stay in your blood system long. And so you can dope up just before you enter that race, and by the time you're done, you basically sweated it out. It's gone. So when they did a urinalysis, they couldn't find it. And so as technology improved, they were able to go back because they kept those samples. And so as in keeping those samples, then they were able to go back and test, and he tested positive. And so, and he's not the only one, he's just the most disgraced because they took, I think he won 12 and they took them all away. So, and you never hear from him anymore, but he's old, he's like me. But keep that, athletes that use it the most, still today, it's easily achieved. Steroid shots, I got one a while ago. So my EPO levels are high. I got one because I'm congested, you probably heard that. So take a look here. As we know, EPO then class low oxygen levels, then your liver and your kidneys release EPO. EPO then gets into your bloodstream, goes to all of your vital organs. And the main thing is, though, it affects your red bone marrow in your blood. The spaces then manufactures more red blood cells. More red blood cells then are put into your bloodstream, increasing your red blood cell capacity. Therefore, increasing your energy. Couple things I want you to know here, especially for the test. Iron is required for hemoglobin synthesis. The element of hemoglobin is iron. Okay? So make sure you know that. It is a substance, and it's absorbed from your small intestines. It's conserved during red blood cell destruction and made available for reuse. Hemoglobin synthesis. Unfortunately, women that hit menopause suffer from iron deficiency, and so they take supplements and things like that, but it can also turn into a positive feedback to the body, therefore being a situation that can be life-threatening for women. Have to watch that. Anemia. Anemia is a condition in which oxygen carrying capacity of the blood is reduced due to deficiency of red blood cell or hemoglobin. So you can have an iron deficiency known as anemia. So if someone has anemia, just they have a red blood cell deficiency. But it could be traced to iron. It could be a sickle cell disease, which runs in the African population, African Americans, through genetics that are here in the United States. They're basically, if we go back to their genealogy, tracing it back to Africa where it originated, it leads to anemia, and it can be fatal as well. especially in third world countries where there's not the medical help as well. So primary causes: decreased red blood cells, hemorrhaging is one. Bacterial infections, blood transfusions, destruction of bone marrow by radiation, certain medications, cancer, viruses, all are reasons of anemia. Malnutrition, defective genes resulting in abnormal hemoglobin structures. That would be sickle cell anemia. That's what it is. Instead of round shape as a disc, it's more like almost like a spear, and it's wicked looking, and it hurts. It hurts. So sickle cell, iron deficiency. These are all types of anemia. And then there's some hemolytic anemia. It's a bacterial infection. Go back and know that, but definitely for your SACS question, no, anemia is a condition where oxygen carrying capacity of blood is reduced due to a deficiency of red blood cells. That's a SACS. That's one of them big ones at the end. And it is one to remember. So no anemia. And here's a good example of iron deficiency anemia and sickle cell anemia. Notice that most of these are round. This would be the iron deficiency on this side. The sickle cell, they're not all round. Notice that they're jagged. Here's where I told you a spear shape. Do you see that? And in the iron deficiency, you can see that it doesn't carry the red hemoglobin. That's the two types of anemia. Everybody see that? Okay, so treating sickle cell is caused by a single DNA-based mutation with codes of hemoglobin. The result is abnormal hemoglobin structure. Red blood cells are sticky, deformed into sickle shapes. Oxygen deficiency. Red blood cells die quickly from this. It can cause extreme weakness and fatigue. Treatments: children diagnosed at birth receive antibiotics. Hydroxyurea is given to causes production of fetal hemoglobin, which is more efficient at binding oxygen. Transport of bone marrow or umbilical cord stem cells can cure the disease. Experiments to correct the mutation in the genome and infuse new stem cells into bone marrow show promise. More technology, trying to eradicate the disease. It goes back to being the vector being from a mosquito that caused malaria. And I don't know the medical thing, but sickle cell protected malaria from destroying or causing the disease in the mostly the African race. So I think it even talks about it, maybe a little bit in your book, so if you get a chance to read it. So the breakdown of red blood cells. After months of bending, squeezing through narrow capillaries, red blood cells lose elasticity and become fragile. They're worn out, damaged, ruptured. Then they're phagocytized. They're engulfed sometimes by white blood cells, but they're engulfed. Hemoglobin separates into 4 subunits. These are types of proteins, okay? Globins chains and with heme groups. Heme groups break down into iron, and this is called biliverdin. Biliverdin. Iron is transported to red bone marrow by the protein transferrin. Biliverdin is converted into bilirubin. Biliverdin and bilirubin are excreted as bile pigments. Globulin chains decompose into amino acids. So this is what happens. So, just a couple of things here. Because of these cell parts being broken down into fragments, okay, you got to remember the hemoglobin carries the oxygen from the red blood cells. It decomposes when this happens. And as it decomposes into heme and globin. Then heme is decomposed into iron and biliverdin. Iron is made available for reuse. And 80% of iron is stored in your liver cells. Okay? So if someone is low on iron and it's stored in your liver cells, what color do you think you start seeing them turning to? Their body will actually start turning yellow because they're low on iron, and that is it's a sign of liver failure. Okay, and so if there's not 80% of iron inside your liver, and that's what they look for, those enzymes, right? Liver enzymes, then that's always telling you something's going on where your liver is shutting down. And our bodies, we can live on a sliver of a liver, which is crazy. And the liver enzymes are looking at it as a way of degenerating. It's really the only organ that we have that can do that. But the liver does so much that we can't live without it. And by recycling worn out blood cells and storing iron. And so if you ever see anybody, especially when you start taking care of your grandparents and even great-grandparents, and you see that they start looking yellowish, you need to make sure they get to the doctor, because that's an early sign they've got they're in liver failure and something's going on. So bile, when we think about our bile, we also have to think about biliverdin is converted into bilirubin. These are what goes into your bile pigments. And your bile fluids go through your hepatic ducts. Remember your bile ducts, hepatic ducts? And we'll talk about that, and they go into your intestines. So what happens to bile fluid? Bile fluid is then basically goes out your fecal net. But when you have a liver problem, that doesn't happen. So you start bloating with bile fluids. And so just like in kidney dialysis, people with liver failure and liver patients, they have to go get a bile dialysis. And normally when you see swelling at the end of life, it's expensive. They don't do that because they're dying, and it's a waste of money to do that because so they'll let them swell up, and usually they're in hospice care. So 80%, what I want you to know here then, globin is broken down into amino acids. Remember, it's a protein, metabolized by macrophages and released into the plasma. All right. And here I just went through that. Make sure you understand this. It definitely has more information here. It makes sense. Hemoglobin breakdown. Here's the life cycle of the red blood cell. I want you to know, class, for a test question, the typical blood cell, red blood cell, lives about 120 days. I do not have it for a white blood cell, only a red blood cell. So in this case, you remember me talking about the bile duct? Here it is, where bilirubin ends up, once everything is broken down and recycled, bilirubin then goes out the bile duct and then dumps into at the duodenum, where the hepatic duct and the bile duct both dump into the duodenum at the small intestines. Okay? And here's the small intestines right here as it goes through then. But remember, hemoglobin, globin, and heme break down into amino acids, iron, and bilirubin. You go through this cycle, and remember, red blood cell comes from your bone marrow. And here they show you a femur bone. Our femur bones give us probably the majority of our red blood cells. And then bilirubin derivates in your blood, in your kidneys. Go through that, know that, understand it. So that process goes on all the time, but remember, your red blood cells, you have millions of red blood cells in your blood count. So you gotta remember, you're losing blood cells every day, but you're also gaining new blood cells every day. On average, you just lose about 100, after 120 days. So now, white blood cells. We're gonna talk more about white blood cells because white blood cells are your defensive system. Yet, in your bloodstream, it's only about 1%, okay? But they do so many different things to protect you. And you have to, so we're gonna go through this and know that white blood cells is for protection. They protect against diseases. We have the word leukocytes is another word for white blood cells. Erythrocytes, red blood cells, right? Leukocytes, red or white blood cells. They have limited lifespans, so they must always be replaced. Don't have any good data about how long they live, okay? White blood cells are produced in red bone marrow, just like red blood cells, under the control of hormones, interleukins, and colony-stimulating factors. There are 5 types of white blood cells. You need to know they're basically identified either being granulocytes or agranulocytes. Okay? Granulocytes have granular cytoplasm and a short lifespan. Over on the board, they're going to have little granules on them. You remember that? That helps you identify them. Okay? The lobes will also help you, but the little granules tell you there's only 3 that have granules. Neutrophils, eosinophils, and basophils. Okay? They have a short lifespan. Agranulocytes do not have noticeable granules. Now, could they have some granules? Yes. They're just not noticeable. There are 2, lymphocytes and monocytes. Now, the largest white blood cell is a monocyte. You remember from the board over there, it's big, isn't it? The lymphocytes is the smallest of the white blood cells. All right, so I got another free question for you. So we went over through the counts of red blood cell counts on, by, actually, just a blood cell count. It was red blood cell count, I'm sorry. Red blood cell count. Remember, and we also gave your percentages, anywhere from 35 for women, all the way up to 50% for men. And I said on average, you know, we know it's 45%, okay, it just depends. But then we talked about your blood counts, red blood cell counts, how many millions there were from women, men. So for white blood cell counts, your average for both sexes, male and female, is 4,500. So put that in your notes. Red blood cell counts were in the millions, weren't they? 5 million for women, or for men, 4.5 million for women. But that would make sense if white blood cell count, if that only makes up 1%, or really less than 1%, 4,500. So if a person came in with 20,000 for a microliters for a white blood cell count, that would tell you, hey, something's wrong. They're sick. Okay? It could be that they just have a bacterial disease, or maybe they got a major sinus infection, and yet their white blood cells are working, or maybe they had an injury, or maybe it's a sign that there's something else going on and they need to do more tests. So put that down for 4,500. So know that they're different in their sizes, and the functions of white blood cell, we're going to go through those real quick. The first one is the diapetesis. White blood cells can squeeze between the cells of capillary wall and leave blood vessels, then migrate toward the infection site. So you remember, if blood cells go from arteries to capillaries to veins, they've got to squeeze in those tiny capillaries. And when they do, and they keep circulating through, they eventually start breaking and tearing apart. But they do this, so diapetesis, that's what they do until they get to the infection site. Leukocytes are directed to the injury site of the proteins called by cellular adhesion molecules. So we're giving you some terminology here. Phagocytosis, remember, is an engulfing. White blood cells clean up bacteria, viruses. They clean up worn out cell parts. And so there is a white blood cell that does that. It's like the medic inside your body. And it's a type of white blood cell. We'll talk about it. But know that they do this. So all of these are what white blood cells do as function in defense from basically these cellular adhesion molecules. So that tells you right there, if it's cellular adhesion, that's part of blood clotting. Okay? If it's phagocytosis, it's part of engulfing. If it's diapetesis, it is squeezing and going through the capillary. It's racing, basically, to the infection site. If it's an inflammatory response, that swells up the area. You ever turned your ankle and it's swelled up? What it does, it contains that area from spreading throughout your blood system. So inflammation is good. That's why they tell you to put ice on it for the first 24 hours to help contain it, because you don't want to put heat on it. If you put heat on it, that can cause the pathogens to spread. Okay? Pathogens don't like anything cold or too hot. They like it just right. So a little heat on it makes them spread. These are called basophils that are help with swelling. Phagocytes are neutrophils and monocytes that engulf. Okay, so we'll talk more about that, but you need to know that that's their major jobs. So white blood cells are attracted to infection sites by chemical released by damaged cells in the process called positive chemotaxis. So chemotaxis means something happened. There's a damage, there's a tear. Maybe it's a laceration in the body. And so then you have all of these different types of functions to take place. But chemotaxis basically it goes to the infection site, but it puts a hormone in the body's blood system to tell the white blood cells, we've got injury. We've got to get to the scene of the injury, and that's what positive chemotaxis does. And it's a chemical released inside of the body. Here's a good diapetesis where it's this they're leaking through the capillaries into the body, and so there's and you can see where it's trying to get through the blood vessel, and how they sometimes have to really get small, or they twist and they turn. That's an example of that. Okay, white blood responds to bacterial invasion. Let's say the splinter punctures the epidermis. You see that in 1. Bacteria is introduced into the dermis. 3, bacteria multiplies. That's in green. 4, the injured cells release a histamine, causing the blood vessels to dilate and become leaky. Okay? You want to keep that stuff leaky, so then the white blood cells can come in, neutrophils then move through the blood vessel walls and migrate towards the bacteria. Neutrophils destroy the bacteria and start engulfing it up. If it's too thick, you can't do it. So if it's leaky, neutrophils can get in there and start, like Pac-Man, start gobbling all the bacteria up. And so histamines are released in your body, and it's a normal type of chemical released in your body's bloodstream that then allows these neutrophils to do their job. So let's talk about neutrophils. We know then their major job is to clean up, right? Phagocytosis. They're small, light purple granules, and they aid base stain. They're 2 to 5 lobed. They're known as the segs, connected by thin strand of chromatin. They're also called PMNs, or polymorphonuclear leukocytes. They're the first to arrive to the infection site. Please mark that down. They're strong phagocytes, means they're the first ones on the scene, and they're the first ones engulfing bacteria. 54 to 62% of these white blood cells, so they're the majority, aren't they? They're elevated when there's bacteria from an injury. And neutrophils do they have granules or not? Back to your nose. They do. They're agranular. Granular. Go back and look. So for the test question here, they're the strongest phagocytes. They're most That's what their major job does. And here they are. Eosinophils then are very similar but much smaller, and we'll talk about it. So let's talk about basophils and monocytes. Basophils are granulocytes, which are large granules that stain deep blue in basic stain. Granules can obscure view of the nucleus. They release histamine to stimulate inflammation. So that's what basophils. Their major job is to release histamine. That histamine then says inflammation needs to be released. They also release heparin to stop blood from clotting. Okay? So blood needs to be leaky at first. You need it to bleed out a little bit, right? Because when it bleeds out, then the histamine stimulates inflammation. The heparin stops the blood from clotting to allow basically phagocytosis to take place, to clean up the bacteria, the many. Remember the neutrophil to do that. It's less than 1% of leukocytes, so these are rare. They're the smallest of the leukocytes. Basophils are. So I gave you the largest, which are granulocytes, or I'm sorry, monocytes. Now I've just given you the smallest, which are basophils. They're similar to eosinophils in the size and shape of the nuclei. Now, remember, red blood cells do not have nuclei, and white blood cells do. All right. So we talked about basophils being the smallest of eosinophils, the monocytes now being the largest. They're agranulocytes because they don't have granules. Okay? They're 3 to 9% of leukocytes. So they're not like neutrophils, right? Being over 50%. They're less than 10%. They only live weeks to months. But they phagocytite as well. They engulf just like neutrophils. They mainly clean up dead cells and debris. They're not really cleaning up bacteria. That's why they're large. They'll come in and get cell fragments. And the word macrophages means they actually leave the bloodstream to become macrophages. That's still a part of phagocytosis. If they phagocytite, it's inside of the bloodstream. If it's macrophages, it's outside of the bloodstream. They're low nuclei, they're oval, and they're the largest. So know the difference, which one's the largest and which one's the smallest, and know exactly what their main function is. And here you can see on the left, basophils, on the right, monocytes. Basophil are granular, monocytes is agranular. So lymphocytes class. They're slightly larger than red blood cells, okay? They're large spherical nuclei surrounded by thin rim of cytoplasm. They're agranular as well. So remember, we're going to talk more about lymphocytes because of what they do. Lymphocytes are agranular, so they fall in with monocytes. Okay? But what lymphocytes have are your T cells and your B cells. they're the attack team. We've already talked about medics, right, being white blood cells. Okay? We've talked about white blood cells causing inflammation, or, so if you think of like a battle, think of a battlefield being the inflammation, okay? Because in a battlefield, remember they have perimeters around the battlefield, so warriors won't try to sneak off. So they get killed too, they sneak off, okay? And so that's the inflammation. The battlefield is the inflammation, okay? And so the medics are there waiting to go out and get and clean up. Now you actually have the soldiers. The lymphocytes are the soldiers. T cells and B cells are the soldiers. So in other words, T cells and B cells are major types, but important in immunity. T cells directly attack the pathogen, the tumor cells. Proteins attack foreign molecules. Remember, proteins are there, but proteins are in your blood, right? And proteins are manufactured by your body, so is red blood cells and white blood cells. So in this case, white blood cells, T cells are attacked, B cells are memory, immunity. They say, okay, they're like the little brother. They stand behind the big brother, duking it out, but they're the ones that have the brains and remember if the virus comes back, if the bacteria comes back. And they say, oh, that's bad, we gotta attack. B cells produce antibodies. That's what they do. So immunity, they come up with antibodies that have memory, and then they come up with an antidote so it doesn't happen again, just like COVID. You get COVID, your B cells come up with immunity, come up with a memory first. I'll remember this next time. So I'm going to come up with an antidote, so if I see it coming, I'll be able to stop it. Guess what? Viruses know how to evolve, change, disguise themselves. Your lymphocytes may live for years in your body. Monocytes only might live for a month. So know that. Lymphocytes and what all they do, make sure you understand it. They come about 25 to 33%. So they would be the next largest under neutrophils, won't they? And they may live for years, and they're your B cells and your T cells. Here are some lymphocytes. Look, even though that they are granular, there's some granules there. They just say you can't see a lot of granules. That's why on the outside coating where you see the red, you don't see many granules, but when you open it up, inside, you'll see some granules. So on your white blood cell count class, I want you to know a couple things here. It's a procedure used to count the number of white blood cells. We talked about, on average, your normal white blood cell count is 4,500. If someone has leukocytosis, they have a high white blood cell count. Anything above 10,500 is considered high. Need to know this. Okay? Acute infections can cause this. Fever. If you have below 5,000, your white blood cell count can be low, especially if the normal count is 4,500. A low count would be under 4,500, okay, on average. Men and women are going to be different. So a person's probably going to look at a low count being 3,500 or less. all right? So keep that in number, but, that is just an average from men and women. Men are gonna be higher, women are gonna be less, children are gonna be less, that's why they come up with 4,500. But they say 3,500 means that there's something going on, and we call that leukopenia. So know the difference between leukopenia and leukocytosis. So examples, measles, mumps, the flu, chickenpox, AIDS, anemia, all are examples. Vigorous exercise, great loss of body fluids, acute infections, acute leukemia would be an example of leukocytosis. Multiple myeloma, leukocytosis. Leukopenia, measles, mumps, anemia, anything below 3,500. Differential white blood cell count lists percentages of types of leukocytes. Percentages may change in particular diseases. Neutrophils increase during bacterial infections. Makes sense, right? Neutrophils get in there. We got a release of histamines. We want to make it leaky, and we don't want to start clotting, and we want to clean up as much bacteria before the clotting process takes place, right? So that's what neutrophils do. Helper T cells, a type of a lymphocyte, decreases in HIV infection. And so the more T cells, the more the attack is going to be. But eventually, a person that goes with HIV untreated, then the helper T cells decline, you can't keep up. And then the white blood cell count drops below, and that becomes AIDS when it gets below that 3,500, and the person is dying, and it's too late. Also, that would be for hepatitis, someone with hepatitis C. But both are curable. Hepatitis C, 5 years ago, wasn't curable. It's curable, it just has to be found, detected. That's why it's good to get it checked. Hepatitis, HIV, it's still not a curable disease, but they're on the brink of causing it to be undetected. And medications to keep you from getting it. Technology is unbelievable. So, a couple of things here, white blood cell population change, elevated lymphocytes, hairy cell leukemia, whooping cough, mononucleosis, elevated eosinophils, tapeworm infestation, hookworm, allergic reactions, elevated monocytes, typhoid fever, malaria, elevated neutrophils, you know you got a bacterial infection, 2 fewer helper T cells, an example would be AIDS. So I've got a SACS question for you. You want to write this down. Infections is an issue for people with leukemia. So people with leukemia, infections are not good. This is why. Immature white blood cells cannot effectively fight infections, causing white blood cell counts to get high or get too low due to the type of infections. Remember, we talked about leukemia versus AIDS. They're both on the opposite ends. Leukemia might be 40,000 white blood cell count, and AIDS might be 2,000. But what they're saying is infections, the helper T cells, there's not enough on the battlefield. They can't keep up with the supply over years and years of fighting the disease. So eventually, it goes into a positive feedback. So, leukemia. Let's talk about leukemia for a little bit. So everybody got that sex question. So in this case, leukemia is a cancer of white blood cells, classified by the speed of the insult. Acute and chronic. Acute means it's discovered at birth or in the teenage years. Its symptoms appear suddenly and progressively rapidly. A chronic begins more slowly, may remain undetected for months or years. My dad found out he had leukemia at 57. He was wrecking the company vehicle, and he would come back and said, every time I'm driving the truck, I see something and I can't hit the brake. My brain's telling me to hit the brake. Well, they found a tumor on his spine that was pushing on the nerves to his legs, so the communication wasn't coming there. Took 3 wrecks, but they found it. They found it, and so unfortunately they had to remove it, and when they did, it caused him to be disabled because they had to cut nerves to his legs and his balance. So just keep that in mind then. So there's 2 types, but he had chronic leukemia. A chronic leukemia means that it's just detected later in life. And normally a person doesn't die of chronic leukemia, but they die of infections. Okay? Lymphoid leukemia, cancer of the lymphocytes produced in lymph nodes. Myeloid leukemia is cancer of granulocytes produced in red bone marrow. So know the difference there. Lymphoid leukemia is cancer in the lymph nodes. That's why they say, oh no, did he get to the lymph nodes? You ever heard that? Mm-hmm. That means it's spread, and cancer is spread. It's left one organ, and the lymph nodes caught it, but it's in the bloodstream. And when it's caught, which it's supposed to do, it slows it down, but it slips on through and goes on to another invasion, to another organ. Treatments include traditional cancer treatments, chemotherapy. Chemotherapy is there to shrink the tumor. Drugs that target enzymes specific to cancer cells. So if you target an enzyme, what are you doing? Enzymes, if they're affected by pH, by heat, guess what? By chemicals, then the enzyme will not speed up the reaction. It'll slow it down. And so that's what chemotherapy drugs do. They're chemicals put in the body to affect the enzyme so it doesn't speed up the reaction. For every molecular reaction, there's always an enzyme to speed up the reaction by just giving it enough. Remember, enough without overdoing it. And what they're saying is, it stops it from doing that. And when it does, it slows the tumor from metastasizing. Does that make sense? And so, class, chemotherapy, it allows the person to live longer. And now it's an available thing. Some people don't want that. They know that it's going to happen. But chemotherapy can slow down breast cancer enough that maybe they can go in and remove the tumor and stop the cancer. Okay? My sister in my dad's side of the family, they all had breast cancer. One died, technology wasn't there. She went through all this invasive chemotherapy, and when they went to do the surgery, they realized that it had spread to the lymph nodes. And so they did a mastectomy, and ended up removing both at the time. And she wishes she just would have had that done to start with, because she lost about 1.5 years of her life going through this chemotherapy that she can't get back. So symptoms of leukemia class: excess number of white blood cells, fatigue, headache, nosebleeds, fever, respiratory infections, bone pain, increased blood clotting, and time causing bruising and bleeding. Know that symptoms, okay? Treatments: traditional cancer treatments, chemotherapy, drugs that target enzymes specific to cancer cells, bone marrow or stem cell transplants also occurs. Remember, they want to take the blood out, let the stem cells, take all the cancerous blood out that's out there, let the stem cells divide, take blood transfusions and try to build that immune system back up of cancer-free. Sometimes it works, sometimes it don't. So know the difference between acute and chronic. Normal white blood cells and blood cells of a person with granulocystic myeloid leukemia. Myeloid leukemia is another blood cancer as well. It's kind of like multiple myeloma. And here you can see someone on the right with myeloid cancer. Platelets, we're going to stop right here. I'll get platelets next week. And I'll finish up this chapter next week going into our midterm. Is that right? Midterm's up next week, is it? I'll have to look. I don't think it is. I think our midterm is the, next week would be like the 6th. I need to look at that, make sure. You guys know? Midterms next week? Okay, I'll double check, but if it's next week, then we will finish this up the following when we come back. Yeah, I'm messed up here.